All right, so good afternoon. We'll get started. As you can all tell, it's virtually springtime, summertime outside, and it's also something.
Closer to your mouth.
How's that? Thank you. Okay, so it's springtime, summertime, and that has many recollections, and I'd like to briefly share one with you as a sort of unusual introduction to today's topic, but I hope you'll see where I'm getting. So this symbol, of course, is the Brooklyn Bridge, one of the two most famous structures in Brooklyn, and one that has lasted. And as those of you who've been coming to this course repeatedly have heard this song over again, but it's sort of the metaphor that symbolizes bridging basic science and human health. I've been handing out wrong information for several weeks. That's actually Manhattan on the other side, not Brooklyn, and the tallest building is the Trinity Church. So for those who called my attention to it, I stand corrected. Okay, now, now this is the baseball season, and it's just started, and it's very exciting, and the other great symbol of Brooklyn is Ebbets Field, which now is a giant housing thing, and there's a little plaque on the ground, and you talk to people, do you know what was here? And nobody has the faintest idea. But this is the world champion Brooklyn Dodgers in 1955. And the next slide shows four of the big stars. There's Jackie Robinson. Next to him is Duke Snider, and next to him is Pee Wee Reese, and next to him is Harvey Alter. I don't know who the fourth person is. Maybe some expert can fill me out. So why should I be showing baseball pictures? And it's very simple. Because the most important person in the baseball team, aside from the pitcher and the coach and everybody else, is the number 4 hitter. Because somebody in the first 3 is supposed to get on base, and the number 4 hitter is the guy who hits the home run. So this is a long track and way of saying that today we have 2 number 4 hitters. And this is no joke. We're really very grateful to both of you for coming, and I hope we all will benefit from exciting new things that we're going to hear. So the first of our speakers today is Jake Liang, who got his medical degree at Harvard Medical School and is here at NIH as the chief of the liver disease branch. Jake has been extremely productive in leading a group that's involved in the exciting developments with hepatitis C virus, which, who knows, may be the first chronic virus infection of man to be cured by drugs. We'll see. We'll hear about it. An enormous problem, and Jake and his colleagues have done outstanding work at the molecular, the cellular, and also at the clinical level. And our second speaker is Harvey Alter, who's chief of the clinical studies and associate director of research in the clinical center and in the Department of Transfusion Medicine. Now, Harvey's very famous because after his graduation from the University of Rochester School of Medicine, he came to NIH as a investigator, as a clinical associate, and played a significant, important role in the discovery of Australia antigen, which was the first clue for the identification of hepatitis B virus. Now, you may not think of it, but in those days, if somebody went and had to have a blood transfusion, the chances of getting hepatitis were very high, particularly if you had more than one transfusion. And there was some knowledge that there were at least 2 major kinds. One you got from water and contaminated food, and that turned out to be hepatitis A, of course. And then there were others that were transmitted through largely blood. And the first one identified was hepatitis B. And Harvey played a critical role in that and applied the knowledge to the blood bank, which then, as we'll hear, eliminated a big portion of this post-transfusion hepatitis. But he wisely recognized that there were other viruses, and then played a critical role in the description of so-called non-A, non-B, which turned out to be hepatitis C virus, which we'll hear much more about later. Now, in recognition of these findings, Harvey has received a Lasker Award, innumerable awards from organizations around the world, and most recently received a very prestigious award from Canada, the Gardner Award. And so Harvey is going to talk about a new virus about which maybe many of you haven't heard. The alphabet is extended beyond D to E and a little beyond, but he's going to discuss the question of E virus and its relationship to the blood bank. When you think of it, the blood bank is something on which, you know, our lives and medicine depend. There's no substitute yet, or it's been a long time. And there's always the question of, does something creep into the blood bank that can then cause epidemics and serious illness among people who get transfusions? Is it possible to predict that? Or if you find something, how do you find out whether it actually causes disease? So these are some of the problems that will be discussed. So thank you very much. And Jake, if you would begin.
Can you all hear me? Okay. Well, Win, thanks for that kind introduction. And when Win showed that picture of Brooklyn Bridge, I wasn't sure what he was trying to do. I thought maybe he was trying to sell the Brooklyn Bridge, but I bet you Win probably could do that. And I'm going to talk about hepatitis C, as you heard today, but I think Harvey Alter is probably much more appropriate to talk about it because he's clearly one of the pioneers in discovering hepatitis C virus, but today he was kind enough to let me talk about hepatitis C, and he'll take on a new adventure on the hepatitis E. So without further ado, I came out with this title, and partly because I felt that since this is a series on demystifying medicine, so I should come up with a mysterious title and then perhaps trying to explain it. And then the second, I think this is something actually has somehow inspired me in what I have done, and which hopefully I'll share with you my thought. As you heard from Win, that hepatitis C is a major public health problem. More than 3 million American people infected with hepatitis C, and globally about 170 people infected with hepatitis C. And HCV is not only the major cause of chronic liver disease in the world, but also one of the few human viruses has been linked etiologically to human cancers. Liver cancer, mostly because of hepatitis C, has the fastest growing cancer death rate in this country. This is really rather alarming if you look at the trend in the last decade or so. As you heard from Win, that hepatitis C was originally known as non-A, non-B hepatitis about 20 years ago, and really took many, many years after that, was discovered in 1989, and the virus cloned molecularly by Michael Houghton and his colleagues. But then again, without the seminal works of our own Harvey Alter and Dan Bradley at CDC, this would not have been possible. And this is a member of Flaviviridae, and has a positive single-stranded RNA genome. And there are 6 genotypes with up to 30% sequence variations. Has a high mutational rates and circulate in the blood in so-called quasi-species. And what's most alarming about this infection, that it can lead to a high rate of chronicity. About 70 to 80% of people infected with this virus will evolve into chronic infection despite active immune response. And also what's dire about this infection, that it has a silent disease progression. Most of the people with this infection don't even know it. And only until they had end-stage liver disease would they become symptomatic. And there's no vaccine available today. And the treatment has certainly improved, but not optimal. I'm going to come back to it, although most of my talk today will not be on treatment, but hopefully you will get some inkling of what some of the research we're doing may have major implication in therapeutic development. The virus has evolved with the host through hundreds of years of ongoing infections. And the virus first infects the host through a series of highly coordinated and sequential steps. First of all, virus gain entry into the cells by interacting with various different host factors, so-called entry factors. And then the viral genome is then released and trafficked to the area of get this working, of ER, in which the viral genome is translated into a polyprotein, and then together with the membrane derived from the ER, forming this so-called replicated intermediate complex in which the viral replication occurs. And then, most intriguingly, through a special organelle in the liver called lipid droplet, that's where the virus assembles. And then from there, the virus then complex with lipoproteins released into the circulation. Alternatively, it could spread to the adjacent cells without going through the circulation. So let me go back to my title. So what is 0 day? And for those of you who know what it is, let me play it out. And so there's multiple choices. Is it your birthday? Is it the day your bank account balance is 0? Or the day you feel your life is a big 0? I think that may be the favorite answer for Dr. Alter, but I'll ask him later. Oh, was it related to the Academy Award nominee movie Zero Dark 30? Or was it September 11, 2001? And finally, was it the day that Ground Zero was named? I think if those of you leaning toward the last 3 choices, you are very close to the answer. And 0 day is a cyberspace lingo. It reflects a vulnerability in the software of a computer that is unknown, but can be exploited by hacker to break into the system. And the most common technique that a hacker uses is called fuzzing. Fuzzing means that you randomly insert data, any random data, into a computer applications to force them to crash. And then what you would do, you would document all the crashes and try to figure out where the crash occurred, and then eventually to fool the computer to reveal its vulnerability for a way to hack into the computer. And this is how most of the hackers can hack into very complicated, sophisticated computer system. And although I'm not a hacker, but this concept certainly has intrigued me. So how can we apply this so-called 0 day for hepatitis C? Again, actually, this is not really new. Such a concept actually has been really proposed many years ago. Gertrude Liang, the Nobel laureate, had made the statement that in the face of great complexity, randomness is not an irrational process. So how can this concept of hacking randomness be introduced in understanding hepatitis C? So this is how I would translate the concept. So if you would introduce so-called random perturbagens, just to perturb the system, into virus-infected cells to force the virus to behave differently, just like you would force a computer to behave differently, in order to reveal its secrets and identify its vulnerability. And I think in the rest of my talk, I'm going to share with you 3 vignettes in which I'm going to apply 3 types of perturbagens that will help us understand the virus much better, perhaps better treatment for the virus. They are small interfering RNAs, microRNAs, and finally chemical probes. So how about small interfering RNAs, perturbagens? For those of you know what sRNAs are, these are highly useful tools that you can specifically silence genes because you can design them according to whatever gene of interest. So using this concept of perturbagen sRNA, frankly, in the lab, initiated a global analysis HCV host interaction by genome-wide sRNA screen. This was published many years ago, so I'm not going to go through detail, but just quickly share with you, this work was done in collaboration with the Harvard Medical School Institute of Chemistry and Cell Biology, who has a screening facility for siRNA. So the concept is basically you can generate random array pools of siRNA representing 20,000 human genes in the human genome, and then transfect them into cells. And then once you knocked out these genes, then you would infect the cell with the virus, and you would 48 hours later you will harvest the cells. The cells you would use either for staining for the viral proteins or staining for nucleides by automated imaging technology. And then the second step, or the part 2, what we call, is take the supernatant from the first step, transfer onto naive cells, and then go through the same procedure. As you can see that both part 1 and part 2 actually looking at different stages of viral life cycles. So using this technique, Frank was able to identify a whole series of what we call host proviral factors that can facilitate HCV infections, or host antiviral factor that can inhibit HCV infection. And this is just an image representation of these staining. As you can see that these are the staining with the viral protein. This is a DNA staining looking at the nuclei. This is a positive control. This is a negative control. You can see that host proviral factor significant decrease viral staining, and host antiviral significantly increase. And these actually can be quantified by automated image analysis and provide you with a database for analysis to identify what's so-called HPF and HAF. So using this technology, as well as combining bioinformatics and data mining, our collaborator at Harvard Medical School was able to generate a complicated HCV interaction map that's shown here. I'm certainly not going to take you through it, but just within one node there, which represent the gene IKK alpha, also called CHUK, is inhibitors of NF-kappa B kinase alpha, represent a very critical intersection point of how HCV interact with the host. So Frank took on an ambitious project trying to understand what's the role of IKK alpha in HCV replication. So first of all, what he had done is to treat the cells with individual siRNA. As I mentioned to you before, the siRNA was used was a pool of 4. So he wanted to test whether these individual siRNA also work. And interesting in that, yes, the individual siRNA works in turn knocking down viral replication, but their potency correlate very well with the silencing efficiency of individual siRNA. So very consistent with indeed the silencing lead to decreased HCV replication. Furthermore, he overexpressed the cells with expression vector containing IKK alpha, and he was able to see significant increase in viral productions in HCV infected cells, as well as production of infectious virus in the culture medium. And in the converse, using IKK alpha mutant, which is deficient in the kinase domain, this behaves as negative dominant construct. He was able to significantly decrease the viral replication of HCV infected cells, as well as production of infectious virus. As you all know, that NF-kappa B is certainly a very important pathway in many biological processes. So there's been a lot of pharmaceutical industry in developing drug against NF-kappa B. So we were able to get several of these drugs that are specifically targeting IKK complex. Two of the inhibitors are active against all IKK. I'll tell you there are 3 subunit IKK, and then but one specific drug will target just IKK beta, not IKK alpha. Okay, so testing these inhibitors, Frank was able to show that these 2 inhibitors, that general internal IKK inhibitor, were able to significantly reduce viral production in a dose-dependent manner, whereas this inhibitor that targeting IKK beta only had no effect. And similarly, in primary human hepatocyte, he saw the same phenotype of these inhibitor in terms of blocking HCV replication. So let's quickly review NF-kappa B. And so you all know that this was discovered by David Baltimore, a Nobel-winning work. This is a process that, in response to external stimuli activate this master switch of kappa B, so-called IKK complex, which consists of 3 subunits, alpha, beta, and gamma. Phosphorylation of this complex will then activate this, which will further phosphorylate this inhibitor of IKK beta here that basically keeping the NF-kappa complex dormant in the cytoplasm. Phosphorylation of the IK beta will lead to degradations, release the NF-kappa B complex in the nucleus to activate NF-kappa B responsive genes. So this is in a short term how NF-kappa B works. And in terms of the virus, what the virus do is that through either toll-like receptors or RIG-like helicase, these are all pattern recognition receptors for infected virus that will also activate the IKK complex. And this is part of the antiviral response involving other factors such as 3, 7, and 9, and leads to induction of the interferon pathway. The IKK beta and gamma are the canonical activator of the NF-kappa B complex. So it's essential, whereas IKK alpha has been implicated by Michael Karen's group to mediate so-called non-canonical pathway of NF-kappa B through the NF-kappa B2 molecule. So in order to explore this further, Frank decided to say, well, let's go ahead and knock out all the NF-kappa B pathways to see what its effect on HCV replication. And lo and behold, when he did the following experiment, he saw that knocking down all the other NF-kappa B subunits actually led to an increased viral replication, which you expect because these are innate immune response and these are antiviral factors. So you knock it down, they will go up. However, on the other hand, just IKK alpha knocking down lead to significant reduction of viral replication. So therefore, we reason that the IKK alpha and other NF-kappa B components have opposing effect on HCV propagation. So how could this be? Furthermore, Frank decided to say, hey, look, since I can overexpress IKK alpha, and that will lead to the phenotype or increased HCV replication. So if we knock down the downstream, if indeed IKK alpha is mediated through this, we shouldn't see any effect of overexpression in IKK alpha. And actually, this is not what he saw when he did the following experiment. When he treated the cells with siRNA against kappa B1 and 2, he still could see the increased viral replication as a result of IKK alpha expression. So it suggests that IKK alpha is going through some other pathway as opposed to the classical kappa B pathway. So therefore, we reason that the effect of IKK alpha on HCV propagation is independent of kappa B. There's also a lot of data that went into proving this because the reviewer really made sure we show them that this is indeed kappa B. I'm not going to go through them, but you can read about it when it comes out in a couple of months that I think this pathway is indeed independent of kappa B. So where does IKK alpha act in the step of HCV life cycle? I showed this to you before. Various different assays have been developed in the last few years to really look at specific step of the viral life cycle. The HCV PP and HCV binding assay can look at entry. HCV single cycle infection assay can look at the early steps. The IRIS assay looking at translation. Replication assay looking at replication. And finally, the HCV CC assay can look at the complete viral life cycle. So applying these techniques, Frank was able to show that IKK alpha actually act on the assembly process. And if you just remind you that the assembly process is intimately involved with lipid droplets. So what does this mean? Pathologically, HCV infection is associated with a pathological hallmark of so-called steatosis. These are the phenomena that hepatocyte, the chock full of lipid droplets, as you see here in this liver biopsy sample of HCV infected individual. Obviously, you have a lot of inflammations, but this is a pathological hallmark of HCV infection. And this is actually very similar to another disease we see very commonly, so-called non-alcoholic steatohepatitis, in which the primary injurious agent is lipotoxicity. This is associated with insulin resistance, obesity, etc. So you can see that this pathologically they look very similar. So clearly the lipid droplet is involved in the HCV infection. And this phenomenon actually has been already described in various different cell culture models. In HCV infected cells, actually it's been shown to produce high level of lipid droplets. As shown here, this is a core staining. As you can see that there's an increased presence of lipid droplets, and they also colocalize well with the HCV core protein. And then when you treat the cells with siRNA against IKK alpha, you can see that the lipid droplet was significantly reduced, although you can still see core there, but there's no longer colocalization of the core with the lipid droplets. These data suggest that HCV infection induced lipid droplet formation and that colocalizes with HCV core. And IKK alpha mediates the effect of HCV on lipid droplet formation. So to further explore the effect of IKK alpha on lipid droplet formation, that Frank overexpress wild-type IKK alpha, as I told you before, that can increase HCV replication. And indeed, when you overexpress these cells, this IKK alpha is tagged with the HA epitope. You can see that in the cells expressing this HA tag wild-type IKK alpha, you see significant increase in lipid droplet formation. Whereas when you overexpress the negative dominant mutants, you see actually a decrease in the lipid droplet formation in the cells expressing this negative dominant IKK alpha. So how does HCV induce this lipid droplet formation? This is the genome of the virus. It's a 10 kilobase RNA genomes encoding a polyprotein here. But of this viral genome, there are two 5' and 3' untranslated regions that play a critical role in viral translation and replication. Furthermore, the 3' untranslated region encodes a so-called pathogen-associated molecular pattern, the PAMP, that can be recognized by RIG-I, binds to it, and activate IR3, will lead to induction of interferon. This was described by Michael Gale's group many years ago. So to make the long story short, Frank was able to show that this PAM was indeed responsible for the induction of lipid droplet, just showing you here. So this is just a staining IKK alpha and lipid droplets. And when you treat the cell, these are not infected cells with IKK alpha. You can see even a constitutive decrease in lipid droplet formation. And when he treat the cells with HCV PAM, you can see that there was a significant induction of lipid droplets, as shown here. But when he treat the cells, HCV PAM treated cells as IKK alpha siRNA, he was able to abrogate the high induction of lipid droplets. And using a different technique of quantifying lipid droplet using both tip by phase contrast, again, HCV PAM was able to significantly induce the green color, that's the representative of the lipid droplet. And similarly, using a viral mimetic, these are the poly-IC that can replicate the function of the many PAM molecules, able to indeed induce the production of lipid droplet. So we reason that IKK alpha is involved in lipid droplet biogenesis, HCV-induced lipid biogenesis that is mediated by IKK alpha, and this HCV PAM is responsible for this effect. So to further study this further, as I mentioned to you that IKK alpha is activated HCV, and Frank indeed showed that in HCV infected cells, IKK alpha was phosphorylated with time, where there's no change in the level of IKK alpha. Furthermore, the phosphorylated IKK alpha is distributed or translocated into the nuclei when he performed fractionation of cytoplasmic to nuclear experiment, and it's just a quantification of the signal, and that's what he saw. So HCV infection resulted in IKK alpha phosphorylation and nuclear localizations. To further explore the localization of IKK alpha in HCV infected cell, Frank performed confocal microscopy to look at the exact location of the IKK alpha, and this is such an image in the HCV infected cells. Red represent IKK alpha, green represent HCV core, so HCV infected cells. And as you can see that in the HCV-infected cells, there seems to be increased red color in the nuclei. And using a Zen software of the Zeiss confocal microscope, he was indeed able to see in these cells infected HCV, there's increased nuclear localization. And actually, he could quantify by measuring many of the mean fluorescence intensity of a lot of these cells to give you exact statistically significant increase of nuclear localization of IKK alpha in HCV-infected cells. Said, and then, so I guess the next question is, so what is IKK alpha doing once it goes to the nucleus? So in order to answer this question, Frank designed the following experiment. He will have 4 conditions. Well, one condition would be just your negative control. And then another condition, just treat the cell with siRNA against IKK alpha. And the third condition would be control transfected siRNA infected HCV versus IKK alpha siRNA HCV infection. So these are the 4 conditions. Indeed, qPCR demonstrate reduction of IKK alpha silencing and decrease HCV replication in IKK alpha treated HCV infected cells. So when he did the experiment and look at the pathway analysis, lo and behold, in the HCV infected cells, many of the genes upregulated belong to the lipid metabolism genes. And these increase in lipid metabolism gene can be reduced by either siRNA against IKK alpha, as shown represented by the blue color. Even in cells that are not infected HCV, there was a significant reduction of lipogenic genes in the cells. And then to look specifically at these 2 lipogenic genes, the SREBPs, as you all know, these are master regulators of the lipid and cholesterol metabolism in the cells. Indeed, when he quantified the expression of these 2 genes, he saw an increase in the HCV-infected cell that can be blocked specifically by treating cells with IKK alpha siRNA. So HCV infection induces IKK alpha mediated expression of lipid metabolism gene, including SREBP1 and 2. And just really to look this further, I told you previously HCV PAN is a responsible element for HCV. Indeed, he saw the same thing, induction of SREBP and abrogation of this induction, the treatment of IKK alpha. And furthermore, when he treated the cell with siRNA against SREBP, he was able to replicate the phenotype that viral replication significantly decreased when you silence SREBP expression in these cells, as shown here. So he reasoned that the HCV PAN induces expression of SREBP1 and 2 and other lipogenic lipid metabolism genes, and the SREBP1 and 2 are important for HCV propagation. So this is just also show that using the overexpression, he can increase the SREBP expression, but with dominant negative mutation, he can decrease the SREBP induction. And again, going back to primary human hepatocytes, using the inhibitors I told you about, they were able to significantly reduce the expression of SREBP1 and 2 in primary human hepatocyte, only with inhibitors against all the IKK, but not the IKK beta specific inhibitors here. So therefore, we conclude that IKK alpha plays a critical role in the transcriptional regulation of SREBP1 and 2. And then just to summarize this part of my talk, that genome-wide screen identify novel host factors required for HCV infections and propagations. One of the novel host pathway in HCV life cycle involves IKK alpha. Unlike the other factors in the kappa B pathway that have antiviral effects, IKK alpha exert a predominantly pro-viral effect. IKK alpha plays an important role in the assembly step of HCV life cycle. HCV infection activates IKK alpha, which transcriptionally induces the expression of SREBP1 and 2, and then activation of lipogenic genes as a result of SREBP1 inductions and lead to lipid droplet formations and facilitate HCV assembly. And then finally, HCV exploits the host immune response and hijacks host lipid metabolism to its advantage. The final point of this is that identification of novel host pathway in HCV life cycle has major implication in HCV therapeutic. I show you a couple of these inhibitors that block the IKK alpha, and as you saw, this phenotype on HCV infection was quite dramatic. I'm not proposing to use inhibitors therapy, but I think this gives you a lead about what you could do in the future. So moving on to the microRNA perturbagens. MicroRNA has received a lot of publicity recently, as you all know, that it's a class of non-coding RNA that's transcribed by POL2 through various different processing that then and export into the cytoplasm, and then in complex with RIS to form this RIS complex that then will specific target messenger RNA for either silencing its translation or leading to its degradation. So how about microRNA in viral infection? There are plenty of examples of that. First of all, many host viral RNA modulate viral gene expression and life cycle. And conversely, that some viruses, especially the DNA virus, encode viral microRNAs, so-called, that will affect the host gene expression. So it's a complicated interaction there. Specifically with HCV, MIR-122 plays a critical role in HCV replication from the seminal work of Peter Sarnow's group published in Science many years ago. And then inhibitors of this MIR-122 is actually being developed as an anti-HCV therapy in the clinic. Several papers illustrate the proof of concept of that, and a recent study published in New England Journal clearly demonstrated the clinical efficacy of this approach. And finally, other microRNAs implicating HCV life cycles via regulation host factor have been identified. This is just some other example. So these are small synthetic RNA, so we can really take advantage of the so-called mimics or inhibitors, tools to explore biology. So if you will transfect the cell with a mimic, that will lead to decreased protein expression of the target genes, whereas when you transfect the cells as these hairpin inhibitor, that will increase protein expression of those target genes. So you can imagine this can be adapted to the high throughput format for gain of function and loss of function, and that's exactly what Frank did, getting a library of about 1,000 microRNA, known microRNA. He did the same genome-wide screen microRNA using exactly the same format, and he was able to identify microRNA that are important for HCV replication. This is just the data that I plotted in this called volcanic plot, and to indicate the cutoff value signal on the x-axis is a fold change as a result of the microRNA treatment, and the y-axis is a p-value. And the colored dotted line represent the fold cutoff of two-fold, and then the black dotted line represent the p-value of 0.0025. So with any point falling this area, considers hits. And interestingly enough, the MIR-122, as I told you about, came to a strongly positive hit right there. And these are just the 4 volcanic plots of different part 1, part 2 mimic inhibitors assay. Again, just point out, this is the same plot I showed you previously, but in the inhibitor assay, the microRNA turned out to be the other side as you expect, because it's an inhibitor that will inhibit viral replication. So this is the map generated by Helen Chart when she put everything together, how all the microRNA interacting with many, many different host factors in a network format. This is certainly preliminary, and many of these host factors actually overlap with the sRNA or sRNA genome screen that I think it lends credence is important. So finally, let me end my talk by looking at the chemical probes of perturbagens. Actually, this project was a beneficiary of the roadmap initiative that was started many years ago using the common funds to encourage people to explore a biological phenomenon using the technology that NCATS, or back then was NCGC, available in terms of looking at large pool of small molecule libraries. So we thought about that, great, we have a high throughput screen assay. So I went to our colleague Wei Zhang in NCGC, say, hey, we have a high throughput assay, let's go ahead and do this. And he looked at my assay format and then he laughed. He said that, no, your assay format, although using 384 well plate and has an automated image, it's not the true high throughput format. When you're talking about screening millions of compounds, you got to really be able to miniaturize even further. I say, oh, gee, that's too bad. So we went back to the drawing board, and Zhang Yi actually spent a lot of time trying to set up or optimize this high throughput, cell-based high throughput assay so we can screen the large chemical library that the NCATS has. So the characteristic of these high throughput assay has to be simple. You can't do staining. You can't do these other things that you do to mark around the cell. You just got to take the plate and read it. And then minimal manipulations, I said, it has to be miniaturized at least 1536 well plate format. This is if you see them, these are maybe contain about 5 microliter of the media in there. And it has to have a very high signal to noise ratio when you read them. It has to have the robust reproducibility. These are very different factors we look at it. Preferably in some kind of quantitative format, so you have a by titration-based approach. And then obviously you got to address the whole issue of cytotoxicity, because when you're doing a cell-based assay. So anyway, not to bore you with the details, we finally were able to set up assays, seemed to work. And then in collaboration with NCGC, we screened about 500,000 compound library, and using 4 doses, using high-capacity robotics and high-throughput luciferase assay, and using various bioinformatics, such as looking at potency profiling, activity clustering, structure clustering, and just really point out one specific interest point. This is salzolidine. This is a compound or precursor of a protease inhibitor that's already in clinical use for hepatitis C. Turned out to be really quite similar with one class of compounds we identified through this hyper screen. So this is really showing to know that we're picking out something very similar to what's actually existing in the clinic. And then you're going this approach, we're able to identify about 600 compounds, and we thought maybe we should go through a secondary confirmatory screen, but this time we will use more doses, and then same format, but similarly we included a cytotoxic assay here. So using this, we'll confirm the activity in many of them, and many of them also were non-cytotoxic. So using this approach, we're able to narrow down to 200 compounds. There's still too many to work with. So we say, so what do we do next? So we decided using a secondary screen, a orthogonal assay that's totally different from our primary screen. Actually went back to our same format that we use for the staining, for the siRNA genomes, because this number is manageable. We can go down 384 well plate. And then using this, we're able to narrow down compound that are high efficacy, high potency, low toxicity, about 158 compounds for HCV lifecycle in the assay. So going through the whole lifecycle assays, we're able to select lead compounds based on high potency and efficacy, no toxicity, targeting defined stage of HCV lifecycle, and also by performing structural bioinformatics, we can understand whether these compounds indeed have drug-like properties, and then have certainly with certain novel structure classes. And so with all these various assays and analysis, we narrowed down to 5 lead compounds, 1 targeted entry, 1 replication, 3 assembly, is selected for SAR chemistry and further development. So going back to the last question, my first question, so are we at 0 day for hepatitis C yet? I think the question is not yet, but we are close. I didn't really have the time to share with you all the development, all the advances in HCV treatment, but as you heard from Win, that we actually be able to cure about 70% of the people with the newest combination of therapy. And just think that we just discovered, this virus was just discovered less than 25 years ago, and we can right now, probably not yet, but probably in 5 years, we can actually cure everybody of hepatitis C. And I think that's a remarkable journey, and it's really, I mean, I think it's one of the things that is very exciting that I'd be part of this history. I'm sure Dr. Alter have a lot to say about that as well. And in light of this, actually, Harvey and I wrote an editorial in Annals Internal Medicine last year on the advancing in the therapy of hepatitis C, titled Hepatitis C, the end of the beginning, and possibly the beginning of the end, and to really highlight what's been happening. But certainly toward the end, both he and I lament the fact that we're going to be so successful, we're going to be out of job in a couple of years. But I think for Harvey it's fine, because he can go retire into the sunset. But I have to find a second career, so I better start thinking about that. And finally, I just want to recognize the people who have contributed to the work in my lab. This is a recent photo of the whole liver disease branch. I'm really blessed to work with all these wonderful people in the group. And people such as Frank, I mentioned some of the names, Veronique Penn, Sid, Krista Murphy, Helen Chard, and Stephen Chu have been involved in the siRNA and microRNA screen projects. and Zhang Yihu, Melissa He, and Virginia Chu and Cameron Schweizer been involved in the Molecular Library Initiative. We certainly want to acknowledge the people who collaborated with at Harvard Medical School in the early part of these studies, and then certainly finally to the NCATS people who've been extremely helpful in moving this project along, and it's really a very extremely valuable resource on NIH campus. And on that note, I'll stop. And I think the plan is that we're not going to take any questions, and Harvey's going to go ahead and give his lecture, and then we will answer some questions together at the end. Okay? All right. Okay. So, Harvey.
Hepatitis E was first discovered in 1981, and it was another bad outcome, sort of the Afghan wars. But this is at the time when it was Russia fighting in Afghanistan, and huge numbers of Russian soldiers were coming down with acute hepatitis. It was an epidemic, and it seemed like it was hepatitis A virus. But when they actually tested for it, it was not hepatitis A. And a Russian scientist, Dr. Liang, went back and studied this further and uncovered a new virus. And as part of his work, he actually ate some stool filtrate with this projected virus, came down with a severe case of hepatitis himself, but was able to get serial samples of serum and stool and everything, and he discovered the hepatitis E virus. So if you want to discover something, infect yourself. That's the lesson today. Anyway, this was now the hepatitis E virus. And it was indeed different from hepatitis A virus, not only serologically, but it had certain clinical features that differed. It had a male predominance, quite striking. It tended to happen in older people. It could be equally severe, but hepatitis A was always a self-limited infection, and there was some hint that this might become a chronic infection. And there was a higher mortality rate. In this particular study, it was 0 in hepatitis A and 6% in hepatitis E, genotypes 3 and 4, which I'll just talk about in a moment. We now know there is actually a mortality rate with hepatitis A in adult patients, but the mortality rate with E is higher. These were 3 studies of icteric hepatitis, and showing that HEV was much more likely to be an asymptomatic anicteric disease, even though it typically presented, it was found because it caused acute observable hepatitis. When you started to sample large numbers of people, you found the majority of cases were subclinical, just like hepatitis C. Hepatitis C is basically a subclinical disease. So hepatitis A was being masked, hepatitis E was being masked because of its clinical similarities to hepatitis A. It was actually probably been around for a long time, but it wasn't until you could rule out A that you could find E. Similarly, reminded me of the recent discovery of the gene for shyness. Now, this gene would have been discovered much sooner, except that it was hiding behind another gene. Well, can't win them all. So, hepatitis E is a smaller than hepatitis C. It's a 7.2 kilobase, non-enveloped, single-stranded RNA virus, and it now has its own classification. It's called Hepiviridae. Like hepatitis C, it got its own genus. There are 4 genotypes, but only 1 serotype, which is important in testing. Genotypes 1 and 2 are actually human viruses. They're endemic and epidemic in the developing world, particularly the Far East. Genotypes 3 and 4 are predominantly swine viruses that now can infect humans. So this is a zoonosis, when a primary animal virus can infect a human. And genotype 3, shown here in blue, is the main genotype in the developed world, the main genotype in Europe and the US and South America. So there are, and the entity hepatitis E, so you have the virus and you actually have the disease, hepatitis E. You can see in the Far East, hepatitis E accounts for a large number, near 50% of acute hepatitis cases. In the Middle East, it's still significant, down about 7%, 17%. In Egypt, about 22%. But in the U.S., at the time that these maps were made, hepatitis E didn't make up any of the segments. The small segment there is actually non-A to E hepatitis, an unknown entity at present. The CDC has recently summarized all the cases that were sent to them. They had a hepatitis E testing laboratory, and people around the country who had cases of non-A, non-B, non-C hepatitis sent samples to the CDC to see if it might be hepatitis E. And indeed, 26 out of 154 cases, or 17%, were hepatitis E infections. And the CDC then studied epidemiologically these cases and found that 11 of the 26 were actually in travelers who had traveled to endemic areas and had genotypes 1 and 2 prevalent in those areas. But 15 were non-travelers who had so-called autochthonous infections. That means the infections developed where they resided. They weren't imported from outside. And you can see the striking difference between those in the travelers versus the non-travelers, where the non-travelers were much more likely to be subclinical anapteric infections, occurred in a high proportion of cases in people who were immunosuppressed by virtue of getting organ transplants. They were all genotype 3. The others were genotypes 1 and 2. And some of them led to fulminant hepatic failure. So these autochthonous cases were clearly different than the imported cases, and that really was a reflection of the genotypes. So genotype 1, the epidemic form, is basically a waterborne infection. It occurs in a relatively equal male to female ratio, whereas genotype 3 is predominantly in males. It's predominantly in older people. Neither one is secondarily spread to any great degree. The source for genotype 1 is, as I said, water, but for genotype 3, as we'll get into, it's food. And the agent of genotype 1 is a human agent. Of genotype 3 is predominantly a swine agent. And there are fatality rates. Strikingly, in genotype 1, it's been described fatality in women who are infected in the 3rd trimester of pregnancy, where up to 20% died of fulminant hepatitis E, whereas with genotype 3, this has not been observed, and the deaths occur in elderly people that we'll talk about. There are extrahepatic manifestations of both. In genotype 1, the pancreas is one of the extrahepatic organs that are involved, but in genotype 3, mostly CNS, different Guillain-Barré-like syndrome has been described, different radiculopathies. There is no known chronicity for genotypes 1, but genotype 3 does now have chronicity, as we'll get back to, in people who are immunosuppressed. So this is the typical course. This slide I borrowed from Jay Hufnagel. This is characteristic of almost all viral infections. where the first thing you find is the viral nucleic acid, here occurring roughly 2 to 4 weeks after the exposure, followed by IgM anti-HEV in the first line going up. Then the disease begins, ALT elevations shown here in yellow, symptoms if you're going to have them, and then later the disappearance of IgM antibody and the appearance of IgG anti-HEV. So this would be the same in hepatitis C or hepatitis B. And also the same in hepatitis B and particularly C is that the disease is really an iceberg. That what we see as severe acute liver failure or even acute icteric hepatitis is a small proportion of this iceberg, is the visible part, but you have a lot of anicteric hepatitis. You have people who have no hepatitis at all. They just have antibody seroconversions or very transient viremias. So most of the disease is subsurface. And again, that's true with hepatitis C as well. So what are the clinical features of HEV infection? Well, the incubation period is 3 to 8 weeks. As I said, most cases are self-limited, but now, very importantly, chronicity is being seen in immune-compromised patients, and that chronicity can lead to cirrhosis. In a cruise ship outbreak, it wasn't one of the that line that's been, what's the line that's been getting in all the trouble? It wasn't one of those cruise ships, but it was a nice epidemiologic study because you had a closed population and there was hepatitis E that occurred on board. It was probably foodborne in shellfish, but it hasn't been proven. But of all the people on the ship who ate that, whatever it was, only 21% became symptomatic. And most of those were elderly men. So I'm not going on any cruise trips. Now, in the autochthonous cases, the average age is greater than 60. Again, male greater than female, at least 3 to 1. The case fatality across all genotypes is about 5%. I mentioned the fulminant hepatitis in pregnancy. And now there are rare cases of acute fulminant liver failure that have occurred even in the U.S. And most of these represent acute on chronic liver disease that we'll get into. Dr. Somalia is here, and she's actually doing a large study of this acute on chronic liver disease, meaning you get an acute something added on to a chronically existing liver disease, and it can become very serious. I mentioned the extrahepatic manifestations, and interestingly, in China now there is an effective HEV vaccine that's gone through a clinical trial involving 1 million people. It was only in China could you do that, and it was highly effective. I believe is beginning to be used in China, is not licensed in the U.S., nor do we actually need it in the U.S. at this time. This just shows the male predominance again and the mortality rates across studies, U.S. mortality being very low, ranging from 4 to 10%. So when you're looking for causes of liver disease now, you have to look for the zebras sometime, and HEV has become one of the zebras, one of the things you typically wouldn't look for. But I also show that because I took this picture when in Africa, and I liked it. So I'm going to give you a case example of looking for the zebra. Here's an elderly man, 79-year-old, who developed fatigue and jaundice. Had no pre-existing history of liver disease, no alcohol use, no other risk factors for viral hepatitis. He had a lot of medical problems, and he was on a lot of medications, many of which I've never even heard of. Importantly, all the medication had been taken long-term. There wasn't any new drug introduced just before his hepatitis. On exam, he was jaundiced, and the laboratory showed a bilirubin of 8.6, a very high ALT level, 1,500, plus the normal level being about 40, elevated alkaline phosphatase. He was tested for hepatitis A, hepatitis B, hepatitis C. They were all negative. Tested for anti-nuclear antibodies and smooth muscle antibodies, looking for autoimmune hepatitis. They were negative. Had an ultrasound, which was unrevealing. So the first thing you do in this sort of situation, though, is assume it might be a drug. They stopped all the medications. And lo and behold, the patient got better. The ALT level dropped to normal, or not to normal, but well, it did eventually get down to normal. Bilirubin came down, so it looked like drug-induced hepatitis. But they were looking for a zebra at the same time, and they tested for anti-HEV, and sure enough, at the same time that the drugs were being stopped, they found that he had IgM and IgG anti-HEV and HEV RNA genotype 3. And as you follow this patient over the course, the IgM disappeared, the IgG remained positive, and the RNA disappeared. A classic case of acute hepatitis E. So since all the drugs had been constant, this was probably the cause of this acute hepatitis. There's been a paradigm shift now in our thinking about hepatitis E, and now I'm beginning to lead into blood transfusion aspects of what the implications of HEV are for blood transfusion. We haven't worried about it until now, but now there's been this paradigm shift because it was always thought to be an acute self-limited disease, so that the likelihood of a donor coming in, asymptomatic donor coming in when there was no long viremia, was unlikely. But now it's been shown, as I mentioned already, that, well, I haven't mentioned this yet, it's found, the antibody to HEV is found in extraordinarily high prevalence in the U.S. I'll get back to that. We now know that this can cause a chronic infection. That totally changes the perception of how serious this disease is. So far, only in immunosuppressed patients, particularly the solid organ transplant patients. It's known that not only can it evolve to cirrhosis, but it can evolve to cirrhosis quite rapidly, much more rapidly than in hepatitis C. And in these immunosuppressed patients, it can lead to a chronic carrier state. Now, what we worry about in transfusion is a silent carrier state. We have not yet shown that there is a silent carrier state in asymptomatic people in the U.S. Evidence for this chronic infection, or just a little bit more about it, is that there have been now multiple cases in liver and kidney transplant recipients, and cases of lymphoma, particularly those treated with rituximab, and in HIV/AIDS. The common denominator of all these cases is immunosuppression. Almost all these cases of chronic hepatitis have represented autochthonous genotype 3 infections in endemic regions, non-endemic regions. The viremia, significant viremia levels, 10 to the 5 to 10 to the 7 copies per ml. This is the scary part, this evolution to cirrhosis in up to 50%, and liver failure and death may ensue from that. So from the transfusion standpoint, immunosuppressed transfusion recipients, therefore, could be at risk, and many of the patients that we transfuse, particularly here at the clinical center, are immunosuppressed. So I worry about this. Now, here's more evidence for this chronic hepatitis. This was a study, a nice study, a multi-center review of 85 HEV-infected recipients in 17 different blood centers. These are organ transplant patients. Out of those 85 organ transplant patients, 56 or 66% had chronic HEV infection. 18 of those cleared when they reduced the dose of the immunosuppression. So again, showing the relationship to immunosuppression. 20 of them they treated, mostly with interferon, and 14 of them went into a sustained virologic response. They got rid of the virus, but 6 remained viremic. 18 were untreated, and of those, 13 had persistent viremia, and 2 of them died from cirrhosis. So overall, 8 out of those 56 patients developed cirrhosis, or 14%. Now, back to this acute on chronic liver disease. This was defined as acute liver injury with jaundice and coagulopathy, complicated within 4 weeks by ascites and encephalopathy in a patient with previously diagnosed or undiagnosed liver disease. So you might have a patient with hepatitis C, chronic asymptomatic hepatitis C, and suddenly their liver enzymes start to go up, they may become jaundice, and they may go into hepatic failure and encephalopathy. Or it could be an alcoholic patient, it could be any other chronic liver disease. So when you see this pattern, you think about superinfection with another virus, a C patient getting B, or a C patient getting E, or a B patient getting E. Or it could be hepatitis B itself that sometimes reactivates in a very virulent fashion. It could be a patient who you've been following and suddenly is imbibing a lot of alcohol. Or somebody who took a new drug or a new herb. Herbs have been implicated in a lot of severe liver disease. So these things that are, quote, healthy can be very bad for your liver. Or it could be a flare in an autoimmune hepatitis or new onset autoimmune hepatitis. So these are the kind of things you think about, but in that picture of acute on chronic, you have to think about hepatitis E now. In Bangladesh, they found in these cases of acute on chronic liver failure that 22% were due to hepatitis E virus. Hepatitis E is becoming a more complex disease than we ever imagined, and a more important disease. And it reminded me of the relative complexity of men and women that I've studied intensely, and is depicted on this slide where we're just a simple on-off switch, but women are very, very, very complex. So I want to talk now about the epidemiology of hepatitis E and the zoonosis. This slide is not completely accurate at this point, but it's, I think, interesting. So a zoonosis is an animal agent that has adapted to humans. And a lot of that is in the food chain. And most important example, perhaps, is this, where a simian immune deficiency virus in primates in Africa got to be eaten by hunters and tribal people, and over time mutated and became HIV, capable of infecting man and then spreading from man to man. The same happened with simian T lymphotrophic virus, where monkeys killed, eaten, adapted, mutated, became human T lymphotrophic virus infection. And the same with simian foamy virus infection. But HIV was clearly the most important of these transmissions. But the other really interesting one is this. Sheep were infected with an agent and caused a disease called scrapie, named because they got itchy and they would scrape against fences. That was shown to be a prion disease. The sheep were then slaughtered and for food, but also part of it, the remains were put into offal. Offal was the food given to cows. This caused a neurologic disease in cows called bovine spongiform encephalopathy, another prion disease. Well, the cows were ground up, including their neurologic tissue, and put into meat pies and hamburgers, particularly in England, and caused variant Creutzfeldt-Jakob disease, the human version of mad cow disease. So this was a clear food chain through multiple species. And there's another disease now in elk and deer called chronic wasting disease, which is another prion disease which hasn't yet been proven to transmit to man, but could through hunters eating fresh elk and deer meat. And lastly, the SARS virus, the respiratory virus in China caused such a epidemic and fear. It was actually traced to a civet cat, and these civet cats were often eaten. I don't know if exactly that was how the virus got into humans, but then it became a respiratory transmitted disease. But these are one way. The things we worry about now, or that cause a lot of our trouble now in the blood world, are these vector-borne zoonoses. So you have West Nile virus, which is really an infection of birds and sometimes horses. But mosquitoes bite the birds and then bite man, and man gets infected with West Nile virus as a zoonosis, and man is the end stage of this disease. But the host is really the bird. And the same is true now for deer and mice. Deer carry ticks. They're kind of like a taxi service for ticks. The ticks bite mice. The ticks then bite man. And these are transmitting Lyme disease and babesia and ehrlichiosis. And all of these are blood transmissible. There's been a lot of talk about using pig organs for transplants, pig hearts or pig livers. And although I don't know of any cases, this would certainly scare me that some pig virus would get into humans. And I should now put the pig up here as a foodborne source, as I'll show you in a moment. So that's the picture of the zoonoses. Why pigs? Well, pigs are the host for HEV, the primary host for HEV. You can show that the rates of antibody to HEV are high in blood donors and go up with age. They're even higher in swine handlers. Anybody who works around pig farms are much more infected. And so how is this happening? How is HEV? We're not generally eating raw pig meat, but there's some things that may frighten you. And I caution you, this rest is not for the queasy. But there is this gastro-elitism movement right now where people eat wild boar pappardelle or pig feet Milanese and a whole variety of exotic foods using pigs that are not always cooked to the ultimate. In Toulouse, France, and other parts of France, there's this ficatelli, which is a tremendous favorite of people in Toulouse, where 80% of the people are infected with HEV. And this is basically uncooked pig liver and other parts. And apparently very tasty. I've come to the conclusion that the French will eat anything, anything except American food. So I found out that pet treats contain pork liver. So have you ever munched on a milk bone? A lot of people try that. There's liver slime, which is, it comes out of pig poop. And it's pooled and then used to irrigate crops, irrigate vegetables and plants. So have you eaten your veggies today? This is conceivably a way that non-meat could be contaminated with pig things. The runoffs of water from pig farms, the irrigation of vegetables. So wash your vegetables. Dunkin' Donuts, I found out, sells pork donuts in China. Scrapple, a very popular southern dish, is made from pig heads and pig liver, and not necessarily cooked to the ultimate. They went around, CDC went around and tested raw pig liver in the supermarkets, just your average daily supermarket, and 11% of these tested HEV-RNA positive. So are every one of these cooked properly when you get them home? Well, the USDA says you have to cook pork meat to 145 degrees, but organs have to be cooked to 160 degrees. And I think people who love organs might not like them overcooked. So that's a potential source. So basically, these are some of the ways, but we really don't know how all this HEV is getting around. But food is thought to be the main culprit. So let's get to transfusion now. This was the first case of transfusion-transmitted hepatitis in Japan. Very, very nicely studied case where the ALT levels are shown in red and the bilirubin levels in yellow. And you can see that about 18 days after a transfusion, the ALT went up to 1,800. The bilirubin went up to 18. It's quite a severe case of jaundice. And they went back and traced the donors to this patient, and they found one donor who had been a repeat donor and on previous donations had tested negative for HEV, but on the index donation given here was positive for HEV-RNA, negative for IgM or IgG antibody, but a 5-month later sample on that same donor showed the donor now to be HEV-RNA negative, but positive for IgM and IgG. So this was a nice case of evolving hepatitis E in the donor, then transmitted to the recipient, and the two had complete sequence identity. They were both genotype 4. So this was a nice case. In that same study, they looked at donors who had elevated transaminase levels, over 500, so quite high levels. And they found that 6 of the 18 who had elevated transaminase were HEV-RNA positive, and 5 or 6 of those were IgM positive. So I think in Japan they screen. Susan, do they screen for ALT? I don't know whether these donors would have been able to donate or not, but certainly it shows that HEV-RNA was in the donor population. Here's the first case in England, a similar pattern except a much delayed onset. This was a patient who had lymphoma, was on chemotherapy about 33 to 40 days after the transfusion. ALT levels went up to around 800. Bilirubin went up quite high as well. And during the course of that acute hepatitis, HEV-RNA was present. Later on, IgM was detectable, and up to this point, IgG was not detected in this case. They went back to the donors here, found that there was a donor who developed flu-like symptoms 14 days after that donation. The donor at that time had an ALT of over 2,000. And the donor was then IgM anti-HEV positive, genotype 3, and the patient was sequence identical with the donor. So these are unequivocal cases. There are about 6 or 8 of these cases now. So HEV clearly can be a transfusion-transmitted disease. There's a nice study in an endemic area. It's harder to pick up transfusion transmission in an endemic area because you don't know whether it's coming from the food or from the water or from the blood. But this, in a retrospective study, they looked at people who had been multiply transfused versus those who had not been transfused. found that 9% of those multiply transfused had IgM antibody and HEV RNA, compared to only 0.8% of those who had not been transfused. When they looked only at those who had been transfused within the preceding 3 months, they found that 43% had HEV RNA compared to 2% who had not been transfused. And when they did a prospective study of 25 people, small study, but they found that 3 of those 25 who were susceptible, they were anti-HEV negative prior to transfusion, 13.6% became HEV infected from the start of the study through the follow-up, compared to none in the non-transfused group. So this is indirect evidence, but it suggests transmission. They did trace these cases, these seroconversions, in the recipients. They traced it to 4 donors who were HEV RNA positive and IgM positive. Here at NIH, Bob Purcell, let's look at some of our old heart study cases. These were cases of post-transfusion hepatitis back in the late 60s, where most of the cases were, in retrospect, hepatitis B or C, and only 1 case appeared to be hepatitis E. I'm going to skip through this time for time, but we've just looked at almost 2,000 donors here at NIH, and we found that IgG anti-HEV antibody was present in 18.8% of our donors. These are healthy volunteer donors. And 0.4% of them were IgM positive. None of them had HEV RNA. You can see the confidence intervals were pretty small because it was a large number of donors. So somewhere between 17 and 20% of blood donors in America are HEV exposed. And that's been shown now in other places as well. Again, you can see in the donors there's this nice stepwise age distribution. It's as if this infection keeps accumulating as you keep getting exposed over time, and the cumulative rate is quite high. The key in our study was a prospective evaluation of 362 transfused patients. So in this donor population where 20% have IgG antibody, what's happening to the recipients? Well, we found that only 2 of these recipients, or 0.5%, had apparent HEV IgG seroconversions. That is, they were negative pre-transfusion and positive post-transfusion. As we did serial samples in these cases, we found no HEV RNA. In retrospective testing, no HEV RNA and no IgM. And we concluded that there actually were no new infections because 1 of them had received a high titer antibody from a donor. This was passive transfer. I'll show you that in a moment. And the other one, in retrospect, probably had infection beginning in that first sample. It was negative, but it was high negative, and probably the infection was starting. So that's that. And so we didn't observe any infections, and the upper bound of that 0 observations is about 0.8%. So very low risk that we have demonstrated so far. This is that particular patient who you can see had no antibody. These are optical densities on the left. There was no antibody present pre-transfusion. And then suddenly at week 36, the last of our prospective study samples, it has a high level of anti-HEV. So you say, okay, this is a HEV infection from the transfusion. But no, if you have the interim samples and if you have the donor samples, you can look at it further. And what we found is that there was no IgM or RNA in the patient, that the patient actually had been continuously transfused, received 17 units of red cells and platelets. We had 69 donor samples out of the 72. What we found was 4 days before we got this sample, he received red cells from a donor who had high titer IgG antibody. So the antibody we picked up was coming from the donor. At that time, he also received an HEV RNA positive donor, And he might have gotten infected, but he died several days later, so we couldn't see the outcome of that RNA positive unit. But it's very important to, and you can only do this in a prospective study, to parse out these cases of apparent seroconversions. So should blood donors be screened for HEV? That's the question we're facing now. It's not a major issue at present, but it's something that the FDA is considering. Susan Zullo is here from the FDA. And what I call this the triangle of transfusion testability. What are the things that make you want to test for a virus? Well, one, can you demonstrate an asymptomatic viremia? In other words, is a donor likely to come in with an infection, but you wouldn't know it? That's what happens with hepatitis C all the time. Is there significant clinical disease? Well, I've shown you that there is if you're an immunosuppressed recipient. Is there proven transmission by transfusion? Yes, I've shown you there is. So with that information, well, the piece we don't know is here. We don't know if that asymptomatic viremia exists. So would you test or not based on this? We also are guided now by what's called the precautionary principle. This came out of the HIV, all the cases of transfusion transmission from HIV. So erudite panels came up with this precautionary principle that said, for situations of scientific uncertainty, the possibility of risk should be taken into account even in the absence of proof, should be taken into account in the absence of proof to the contrary. Anyway, our typical way is to wait for evidence-based medicine to say, yes, we should be doing this. But the evidence base often doesn't appear for years and years and years. Meanwhile, people can be infected. So the corollary of the precautionary principle is that measures need to be taken to face potential serious risks, even without absolute proof, unless you have proof to the contrary. So that's in the background in making our decision also. So why aren't we doing HEV testing right now? Well, one is we have no standards. We have no pedigree panels by which to compare assay sensitivity and specificity. If you come up with a test that has high non-specificity, you start to eliminate millions of blood donors. It's a very dangerous thing before you have good assays. We're not at that stage yet. We're probably going to have to screen for HEV RNA rather than antibodies, since antibodies are so common. We don't have any HEV RNA assays that are currently licensed. I don't know if any are in the pipeline. This is the key here. We don't know the frequency and the duration of asymptomatic viremia in immunocompetent blood donors. And this is what's going to drive the risk equation. If it exists, then we have a risk. If it doesn't exist, we don't have much risk. It'll be more like hepatitis A. We don't know the minimal infectious dose and the frequency with which that dose might be exceeded in healthy blood donors. And we don't know the frequency of clinically significant infections in immunocompetent and suppressed patients, though we have a good idea that it is significant in suppressed patients. But how much disease and how often would it occur, and what's the cost-benefit ratio of implementing a test like this? So those are the things that blood banks are dealing with right now. So I'll skip over that. But just to say that this is not unique to blood banking. This is not unique to HEV, rather. There are many agents that we don't currently test for because they're too prevalent or not prevalent enough or whatever. But basically, any agent that has an asymptomatic viremia is a threat to be transfusion transmitted. And the likelihood of that transmission is highly dependent on the duration of the viremia, and the concern is dependent on severity of the disease that it causes. So in this case, we have variant CJD, we have malaria, we have HHV8, we have dengue, we have many tick-borne diseases, encephalitis diseases that we don't test for. We can't test for everything. So we're on the vigilance. This was another of my African slides. but it's not a unique dilemma for HEV. We've conquered other problems. We've handled mad cow disease. We don't worry about these mad cows so much anymore. We have measures to prevent that. We've gotten past the threat of bird flu, although it caused devastating disease in the flamingo population. These are not real flamingos, folks. This is Florida. This is Florida, where these flamingos are standing on every lawn, propped up. So I see no reason why we're not going to be able to handle these sweet little pig things. I think we'll get to this problem, and we won't test, but we'll know in a couple of years or so. So in conclusion, I agree with everything I've said, and we can have questions now for both of us. Thank you.
Thank you very much. That was awesome. So, Jack, you want to please speak up, and there are people listening, so I'm sure you're talking to a microphone.
Well, I don't think your mic's on.
Yeah.
When?
Why the hepatitis virus target the liver? How or?
Okay, well, that's a good question, because they are very, I mean, they do go perhaps to other organs, but they're very specific for the liver. And it's a matter of receptors. I'll let Jake answer this better, but I think they're the specific group of receptors that the virus needs to invade an organ are present in the liver. And
Yeah, in addition to the receptor or the entry factor response to the virus getting in, they also factor that liver-specific, that important for viral replication, assembly, etc. So there are other factors of relevance to the liver-specific tropism.
Yes, here.
I have a question for Dr. Walter. The case that you showed. Thank you. I have a question for Dr. Walter. So the case that you showed where blood transfusion caused seroconversion in that patient, you said he died, but do we know if liver failure was a reason?
No, he died of his underlying disease. It was a very sick patient. He had received 72 units of blood. He was in bad shape. It wasn't acute on chronic.
So is there a move to test, even given the difficulties, if an immunocompromised patient is going to be receiving blood transfusions, is there an effort to select out that group to test the blood that they would be getting? Or?
That's hard to do. It's hard to have two inventories in the blood bank. It was done in the past where CMV screened blood only went to immunocompromised patients, but I think if the tests were implemented, it would be done for everybody. But your question raises the point. If we want to study this further, maybe what we should do is study immune deficient patients at NIH pre and post. We're actually doing that to some extent, but specifically target immunodeficient patients to see if they are converting.
And what about in the Middle East and other parts of the world where you showed the data of the very high prevalence of this? What are they doing?
I don't think anybody is screening for HEV for the blood supply, and the higher the prevalence, the more difficult it is to screen, actually. But if you screen for antibody, if you screen for HEV RNA, that could be done. But in those areas, most of the transmission is coming from water supplies in the undeveloped nations, and in the developed nations, the rates of new infections are low. So it's a lot of effort for maybe a small payoff. But I think the key really is to first to get the standardized panels and standardized tests, because we couldn't do this even if we wanted to right now. And secondly, to really study the asymptomatic viremia better. By the way, the Red Cross is testing 10,000 donors to see how many have HEV RNA.
You mentioned that there was a vaccine available in China. Could you talk about its efficacy, and is there interest or intent to bring that over to people who are diagnosed with HEV here?
Well, the efficacy is very good. It's about 95% efficacy. And I think in places where HEV is very prevalent, this could be introduced, like China, could be introduced as a national policy. But it's not a therapeutic vaccine, so you don't want to give it to people already infected. So the need in the developed world is not sufficient right now, I don't think, to introduce that vaccine.
So what about injection drug users for HEV? Should they be vaccinated for HEV?
No, I think we should vaccinate the overeaters.
And just
Plug overeaters, yeah.
and just one other question: if someone is HIV positive, but their disease is under control with ART, are they still more susceptible for HEV infection?
No, I doubt it. I think it's all a matter of the degree of immunodeficiency.
While I'm walking up here, what do the mitochondria look like in HCV? Would you have lipid droplet accumulation?
Yeah, I mean, we haven't really looked at it closely. It's possible they may be infected. And there's some data suggests that mitochondria may be involved in sort of HCV replication, although it's not clear how that is involved. Maybe it has something to do with the reactive oxygen species and etc. But it's not directly involved. But there may be tangential effect of mitochondria's functions on HCV that remain to be shown. So.
In blood transfusions, what is what is known about optimal matching of the donor and the recipient? And the second part to the question is, what are the measures of success in blood transfusion?
Well, I mean, the matching issue is a different issue than transmission. I mean, we match donors for many different blood groups, not all, and we look for antibody to blood groups in the recipients. But as far as the transmission of the major diseases, HIV, HTLV, HBV, and HCV, those are now extremely rare phenomenon. HIV, HCV, about 1 in 2 million risk of being infected from a blood transfusion. So the blood supply is really safe for the agents that we can test for. So the risks of blood transfusion now are either going to be around the agents we don't yet test for, or new agents that might evolve, or things that have nothing to do with infection. Right now, you know, there's bacterial contamination of blood products. There are all kinds of other issues in blood unrelated to infectious disease. But as far as infection, we've come a long way, and the blood is incredibly safe.
What's that mean? Is the RNA positive, IgM positive, IgE positive? What's that mean? Is it protect, or the people get the protection, or
no?
Or
if
the infection?
If you have HEV RNA, you are infected at that point, and IgM is also a sign of recent infection. If you have only IgG, there may be some level of protection, but I don't know that. But typically, IgG alone is protective, although not in hepatitis C, it isn't though.
Harvey, what's the status of treating blood in advance without knowing what agent may be present.
Yeah.
Purifying the blood, or whatever the
Yeah,
term is.
this is the holy grail of blood transfusion. There are technologies, two of them. One is sorolin treatment with UV light, and the other is riboflavin with UV light. They can kill a lot of agents, but not always get you down to 0 copies of that agent. In Europe, in much of Europe now, they're testing platelets. They're using one of these two methods to, quote, purify platelets. Seems to be effective and seems to have no deleterious effects. The U.S. FDA has not yet approved that process in America. But that's where we'd like to head. It hasn't happened yet. The biggest impediment is red cells, because if you add agents to sorolins to red cells, you kill the red cells. Or you might kill the red cells. Riboflavin seems to be safe for red cells.
Is there an increased risk or severity in pregnant women for hepatitis E?
I don't think so. I think it's only in Asia, and it's if you are in the last trimester and you actually get the hepatitis at that point. I don't think they're more susceptible. They're more susceptible to bad outcome, but not to the infection itself.
I don't understand that.
Yeah?
I don't understand that.
No, if you're
Susceptible to
a
bad
pregnant
outcome.
woman, you're not more likely to get the infection, but if you get the infection,
Oh, I
you're
see.
more likely to have bad outcome.
Well, listen, do you want to have a last word? Anybody? No? I want to thank you both very, very much. This was extraordinary.